And then we talk about phytoremediation. This is when you use things like plants to do this work. And then you analyze, then when you can just go there to analyze what, is it polluted? How polluted is it? What species of meadow is there? So there's a whole area of things that we can do there. So when I like this, and I wanted to put that title there, that quote there from Werner van Braun. So if I do research, I really don't know what I'm doing. Okay? So I just want you to know that. When I'm doing research, I really don't know what I'm doing. But that's why I'm doing it. Okay, let's move on. So let's start with composted sludge. What is this thing? So composted sludge is when you go to the bathroom, which we call washroom sometimes, right? Or toilet, as we used to do. Unless you're using a pit latrine, you're going to flush it, right? We flush our toilets, right? Or wash it down. Where does it go? None of us concerns ourselves with what is next, right? But ideally, when you flush that, it goes down to the municipal, and then it goes to the municipal treatment works, right? And there, they always, if you have been to one of those, they have lagoons, they have all those ponds, and there's a lot of bacteria. They have bacteria that's going to eat that. So there are many, many processes that it goes through. At the end of the day, or at the end of the process, after several days of curing or whatever you do, you have a separation between the solid and the liquid, depending on the city and the size of waste that you have. If it's liquid, many places they have what we call UV light. You all know UV light can be used to what? Kill pathogens, right? So some places like in the city of Jacksonville where I was, it was a smaller city, they would use UV light to kill any pathogens that are in the liquid that is going back to the creek or to the liver. But what happens to the solid? It's a big cake, it's a big mud, if you want, that is now left back in the lagoon. And so they bring scorers, and they bring, for lack of a better word, tractors and caterpillars that get all that stuff and put it on what? Trucks. And they will take it to some dump site. But now there should be a better way to do that than just dumping it. Okay? And so some companies now said, hey, it's there, it's available, it's cheap. What is in there? Can it be? And so they actually packaged these, and now it's sold as compost manure that you can use for your lawn and flower gardens. And so that is the material that we are talking about here. So you'll go to the store, to the Nivers, for example, and you'll find a section that is for gardening, and you'll find that bag of manure, and you take it home so that you can use it for your flower gardens and lawn gardens. And so the question comes, when you apply this material, what is it doing? First of all, what does it have? Is it introducing the heavy metals, or is it actually uptaking the heavy metals? And so this is the study here. So the motivation here is that as we have more population, we're going to have more waste. We're going to have a lot of this stuff coming up. So what are we going to do? Can we use it better than just dumping it? And if it has heavy metals, who cares? Why do we care about the heavy metals? Of course we care about the heavy metals, because depending on what metal you're talking about, lead, which comes from the paints and the bullets and all that stuff, cadmium from cigarettes and all places, chromium from all that, you can see your brain, your kidneys, your lung system, your liver, all those guys are coming in. And cadmium is well known to be one of those that causes lung cancer. So that's why a number of smokers end up down the years to have this problem. So yes, we have to have concern, and I only highlighted these three, but there's a whole cross section of heavy metals that are lodged in that stuff that you might want to concern yourself. So how do we do that in the methodology? So we are moving on in the methodology. Next slide. You take that stuff. I think we are stuck. We're supposed to move on to the next slide. I knew we would be stuck somewhere. Normally, we would have a small remote that you could do that, and we didn't organize ourselves there. Okay, thank you. Now, so in the methodology, you would take that stuff. It could be a little wet. You would dry it, right? And then when you dry this stuff, it's still kind of like, salt or silt. You want, as a scientist, of course, you want to sieve it because you want to know what particular size you're working with. And then when you sieve that, then you'll take a given, I always tell my students, your grandmother is always doing chemistry. The only thing is she doesn't measure it. But because we are chemists, you take a particular amount of it and you mix it with the metal that you want to work with, and you would work at the various concentration of the metal because you want to know. You want to have parameters that you are gauging to see how it works with those parameters over time. And so in this case, we just took that, exposed it to given concentration of heavy metal, and you do that for a given time, and then later on we'll vary the pH and look at how the pH affects that. And then later on, and when you would, in the environment, what makes the environmental chemistry different from the lab is that in the lab you have a test tube, you have a beaker, you have control of what you put in. So the conditions are kind of sanitized and controlled. In the environment, you don't have that. So if you bring lead here, there could be copper, there could be chromium, there could be what. So you also have to look at the effect of what happens if we have other elements there. Is this stuff going to work? So that's what you see there. And a couple ways you can do that is you can do it in a batch form where you put it in a beaker and shake it up for 24 hours. Or you could use a column where you are actually running the solution through the material that is packaged in a column. And in both cases, you want to see what's really happening. And then you would use, most of the time when you're dealing with metal analysis, you want to use the atomic absorption spectrophotometer or the graphite furnace AAS, or you can also use the ICP OES or ICP MS. So in this case, we're using the AAS. So the next slide will give you the idea that now, what did we, you can tell, I am not giving you all the meats. I'm just moving from space to another to kind of give you an idea of what was this. So in this case, what did we find? Well, the table shows you the binding capacity. I don't know if that's clear, millimole per gram of compost. So what we are saying here is when I take a gram of that material, remember, you put the material down and you want it to uptake. First of all, is it uptaking? Look at it. Is it going to introduce or is it going to uptake? We found out that it actually uptakes the metals. In other words, it's, for lack of a better word, it sucks it up, right? And complexes or binds it. So what you're seeing there is breakthrough curves. Those breakthrough curves are telling you that as we continued to load means apply or increase the amount of lead on it, it took time for you, because what you're doing is, if it's a column, you are loading, you have a column packed with the material, and you are loading lead through the column, and down at the bottom, you're collecting. I'm putting here, I'm putting 100 millimolar, ppm, sorry. If I'm putting 100 ppm of the lead, I expect, if nothing is happening, to collect 100 ppm. And when you are analyzing the liquid coming out, which we call effluent, you find out that from your 100, you are not getting anything. So a lot of that has been what? Absorbed. So you do that for a long time until you get a breakthrough at around that point there, 15. And that's for one element. That's for lead. And for copper, you see the breakthrough is around 2.5. And for cobalt, is almost an immediate breakthrough. That is just telling you that the material has a high binding affinity for lead and a very low binding affinity for what? For cobalt. So that is, I'm giving you again examples of the metals, but we did quite a good cross-section. So then what comes out there is to tell you that lead, then iron, then copper, then zinc, then nickel, and then cobalt. So that's what you get. And the table there tells you that for 1 gram of that material, if you were to expose it to those metals, lead will take 0.091 to the gram, and down the road where manganese is almost nothing taken in. So the material will be very, very good for use for those fast metals there. And then the key question comes, what is the effect of pH? If I'm doing this at low pH and I'm doing this at high pH, how does this affect the ability of the material to uptake the heavy metal? And of course, and the next question is, okay, you can bring something and you can hold it. How firmly are you holding it, right? If we are using it to clean it, we want to be able to show that it took it up, and the rain is not just going to wash it away, because then we wasted time, isn't it? So that's part of the question we have to answer as scientists. How firmly is it held? And so in the next slide here, you see the effect of what? The effect of presence of other metals and pH. You see that? So when it comes to presence of other metals, what you are seeing is, now I'm learning lead with other metals presence. If, for example, I have the ratio of 0 to 1, it means on that first one, the one that has the dots here, that is, has a longer breakthrough, that's when it's only lead. But when you introduce 0.1 of iron, you see how you begin to reduce the binding, the uptake ability, up to the point where you have 1 to 1. You can see how much it has really it's reduced it. And when you have more iron than lead, then you totally really reduce the ability of the material. So what's happening there is a competition. There are many processes that are responsible for that uptake. It could be a complexing, it could be an exchange process, right? And it could be an interstitial into the residual part of the material. But it is telling us that these two metals are competing quite effectively in doing that. And so that is what that tells us, that if you have the place polluted with iron, the lead uptake may not be as good if the concentration of iron is high. And then what about the effect of pH? Again, you can see that now we are varying the pH from acidic to what? Well, low acidic to slightly mild acidity. And somebody would say, why didn't you do that in the basic pH? In fact, if you have done a little bit of environmental work, you realize that most of our waters and most of the areas where this pollution happens, things tend to be on the acidic side. And so that is why we worked on this. And you can see that when you are at low acid, pH 2.19, you have minimal, very minimal uptake. And when you are at mild acidity, then you would have a large intake here, that uptake of the heavy metal. And again, that tells you that you want to do your things. So if you had an environment that is highly acidic, you probably want to neutralize it before you use this process. But most of the environment is going to be pH between, in fact, that's what we find in the waters. You'll find it between 5.5 and 6.5. So this process is going to work well. So the higher the pH, the better it works. And of course, if you're dealing with other metals like iron, if you go to the basic side, something like iron is going to hydrolyze, right? And it's going to form iron hydroxide, which is insoluble, which is going to be slightly different in this kind of process. So this tells us what happens with the process in terms of this. And why is it that acidity is affecting this uptake? Again, remember, we are dealing with metal ions and acids, so there is that competition between the hydroxyl ion, not the hydroxide, but the hydrogen ion, which is also the hydronium ion, with the metal. They are both on the positive, they are both going to compete, and sometimes size comes into play. And so that is why you can see that ionic effect, or a competition between the metal and the ion. So how strong? So you can't stop before you figure out how strong the binding is. So if it's doing it, is it holding it strong enough? Now, Tessie and others have, in the next slide, Tessie and others have done work that will show you. Again, the next slide, please. Tessie and others have done work, and they have come out with a methodology that would help you kind of extract what you bound to the material from that system. So in this case, it's like you do it in 4 or 5 phases. And so you have the first time you take that, you run acetic acid. So you take the same material, okay, and then you have already loaded the metal on it. It has bound the metal. Now you want to see how you can leach it off, take it off. So the first part is to use acetic acid, and then you run that, shake it overnight, centrifuge, filter, take the residue, and then when you take that residue, put the next extractant, which will be hydroxyl ammonium chloride, then do it again, shake it, rinse it, and then the next step will be the hydrogen peroxide, which, as you know, is highly oxidizing. And then you do the same thing, shake it and do that. And then the last step is to use aqua regia, which is really strong acids. Now you're dealing with hydrochloric and nitric acid, and then you shake it, and again you shake it and filter it, and then you get the effluents. So in every it's sequential. You start with the low ones that will mimic rain or acid rain, all the way to the strong acids that are going to extract anything that is in there. And then you want to see, where was my metal lodged? Okay? And so in the case of the lead here, I'm showing you before we load the lead, the much we could get out of it, the material. You could only get 0.014, and then you keep going, and this is part per million, and then you keep going until you see most of it could have been, some of it could have been in the carbonate or the residual fraction. But now, after you load it, you want to know where it is. You can see that the exchangeable fraction is about 30%. The exchangeable is the one that is easily exchanged, ion exchange. But you keep going, you see there is a little bit that is absorbed that will be washed with water or some mild acid, right? And then organically complexed. Now remember, there is complexing and there is bond forming. We're not talking about bond forming. We're talking about what? Complexing, complex formation with the organic material. And you can see there is about a small percentage there. But you see the larger percentage of lead is in the what? Carbonate fraction. And then we have some in the residual that is removed by the extraction of the strong acids. So the beauty of this is telling us that 70% or more of the lead is held in the carbonate, organic, and residual fractions. That tells you that that material is going to hold that lead no matter what kind of rain you're going to put through it, which is good news. Okay? And less than 30% could be exchanged easily, and less than 1% is loosely held or could be washed with water. So what do we say here then? Then the take-home message becomes that the composted sludge is a very environmentally friendly way to remediate these metals. Well, is that the only thing we can use? Not exactly. What else is cheap and very ubiquitous? In other words, so much around and so much cheap. Well, make your guess. I'll help you. Right now, I live in America. America lives on coffee. Okay? They thrive on coffee. I don't. I do what? I thrive on tea. Now I'm drinking my tea. Apart from water, tea is the world's most popular drink. It's number 2 in the world. Okay? So coffee is in few places. It's the second most popular drink after water, about 5 billion cups a day. In fact, that's the minimum. I saw some literature that says it's about 12 billion. I didn't believe that one. So I'll go on the, I'll go on the conservative side and say it's 5 billion. If it's 5 billion, it means we are talking about 5 billion grams per day of coffee. 10 billion grams per day of coffee, and 10 raised to power 4. That's 10 what? 10 raised to power 4. Come on, help me there. 100,000 tons, right? 100,000 tons of grams of tea per day. That's a whole lot of tea, right? Yeah. So the funny thing is, yes, you drink the tea. Either it's the tea bag or it's loose, whichever way you make it, right? If you are using one bag, it's say so many grams, right? What do you do with the bag after you drink the tea? You know? So I have the bag here. So if I take this bag, tea bag, and I put this in water, right? It's going to seep through. I don't eat the bag. Do you eat the bag? No. What happens with what is in the bag? Right? Goes to waste. So you waste almost 100% of the solid, don't we? So that's a lot of solid there. So you're talking about 90% of the tea becomes waste. Again, almost 100,000 tons per day, and most of that is discarded and nobody thinks about it. So could it do something? You betcha. Of course, there are many types of tea, whether it's black, green, or oolong. How many know what oolong tea is? Anybody ever drank oolong tea? So the difference between green, black, and oolong is the processing. Green tea is least processed. Okay? If you go to the tea research or where they do tea, they would actually have green tea and black tea. Black tea is the most processed, is the most oxidized, is the most stable, but it's the one that has the least amount of antioxidants. So if you want to really reap your antioxidants from tea, you want to drink green tea. Oolong tea is also mildly processed, so it's the second better cup of tea. And then what we drink most of the time, which is black, is really the most oxidized and the least beneficial. Why do people sell mostly black? Because it's the most stable. It's going to last longer in the shelves or at home in the cupboard. So you buy that. And it's also cultural. So again, this is a very noisy slide. The next slide is very noisy, so which is going to be the method. I think I'm ahead of the game here. I'm sorry that you missed all the things. I think my tech, you really have to catch up with me, please. I'm talking assuming they are seeing it, and they are not. So I'm already done with this slide, but this is what I was talking about. I knew this lag was going to be a pain for us, and I'm sorry about it. So the next slide here is talking about the process of the tea. So we're going to brew, and again, we are thinking about tea waste here. So before you get to the waste, you have to brew. Okay? So we're talking about you brew the tea, but here we are brewing it in bulk because we are just interested in generating a lot of waste so we can use it for research. So you brew the tea, and then you do a similar thing to what we did with the compost. You take that tea waste, you dry it, you sieve it to size, and then you load it with heavy metal. Again, you can choose any metal that you want to load it with. And so in that case, we are talking about loading it with lead and all that stuff. And then after you load it, you again going to see how much it's bound. You're going to do what we call metal leaching, or extracting that metal out of it. And of course, you use the same techniques of atomic absorption spectrophotometer. So next slide here. What do we find? And again, I'm jumping A to B because of time. In this case of tea, again, we realize now, if you look at this, I'm showing you 2 or 3 things. One, cadmium, chromium, and copper. Now, I couldn't show everything. I just chose these ones as representative metals in this slide. And I'm showing you different particle sizes. So we are talking of 300 to 400 micrometers. This is the particle size. So if we want to talk about micrometer, you're thinking of that particle size as if it's a diameter. So we are talking of 300 micrometer. That means 300 times 10 minus 5 of a meter particle size. So that's the smallest, and the 600 to 800 is the largest. So we did this to see the effect of particle size on this uptake. And as you can see, they compare favorably, but the middle size, which is 425 to 600, is a little better. So we stuck with that, and that's what we used for most of our studies. And also we found out that lead better than cadmium and better than chromium, and then copper when it came to what? The binding or the uptake. So T-waste was preferentially taking more more lead than cadmium than chromium, and least copper was held in there. And what about the pH and the pollution effect and the mechanism of uptake? So here we just did we did a competition with so many metals together, but I'm showing you an example here of cadmium and copper. And in this case, you can see that cadmium preferentially much more than copper, and you see that at the higher concentrations where you have say 100 parts per million, you had that uptake for the copper. But when you are at the low, which is 5, both of them showed favorable preferential uptake. So how strong am I ahead of the game again? Yeah, sorry, I'm not used to telling the tech to move the slide. So this is what I was talking about. The copper and the cadmium, you can see at the low concentrations, you have both of them very comparable. But as you go to higher concentrations, say 100 parts per million, you can see that cadmium preferentially more bound or held than the copper. Next slide. The mechanism, how strong are these metals held by the T-waste? So again, you can see here, what I'm showing is, I'm showing copper, cadmium, and chromium, and I'm showing the various fractions. I'm showing the chemicals at the bottom, but the chemicals are representing the exchange, the organic fraction is represented, for example, by that peroxide there, and then you see the last part there representing the leaching off. And you can see that the HCl, HNO3, which is the aqua regia. So you can see the beauty of these is showing the superiority of T-waste in binding these metals. And you can see that in the case of copper, the larger fraction, almost 80% plus, is in the organically complexed and residual fraction. So it's very hard to get it off once it has bound it. And you see, even better for cadmium, a larger percentage is in the residual, which means you're going to take very strong acids to remove it once you have bound it to the T. And in the case of chromium, behaved similar to what? Similar to copper, but you have a little more chromium lodged in the residual fraction. So this is very good news for use of T-waste in the uptake of these heavy metals, because not only are they going to take them up, a large percent, they are going to hold them firmly, which is what you want. So what's more? We've seen we can use biosolids to clean up the environment, especially when it comes to heavy metals. Is that all I do? No, I do a whole lot more with the environment. So I'll let you into a little bit of else that I do. So the University of Lavana has a Montana field station. Montana is one of those Midwest states that is so far in the north, is a neighbor to Canada. So we have a station there, and I went there, or we send our students there, and in that station we collect samples from that area because of some of the environmental stuff that goes on there. One of the things that goes on there is that there was a Superfund site where there was a big mining in almost 5 to 10 decades ago. And that mine is there, and they try to clean that up. And part of what you do as a scientist to make sure, is it clean? Is there something happening? Anything still leaching out of it? And then, of course, the phytoremediation that I talked about. And I also do look at soils. And but let me see what I am going to show you in the next slide. In the next slide, it's what we did some of the work in Montana. So it's a very busy slide. I don't know if you can see the map. Please, by a show of a hand, can you see the map? Yeah, thank you. So the map is showing you Montana, just a little section of Montana. And down there, if you see something called Buckley Pit, right there where there's a thing down there, Buckley Pit, that is where they have a big, you could almost call it a well or something, where they used to do the mining, and it's now like a pond where they did a lot of copper mining. It was one of the largest mining areas there. And when they mine, they don't just mine copper. They mine copper with many other minerals, and so. But there's a creek there, and I don't think that the creek is showing very well. But this is the Anaconda. This is the Anaconda Creek that runs through here and branches out here and goes over there. So what you see as numbers, which don't show very clearly, those are stations where my students went and they were sampling out. And so you sample along the way and also along the creek. Why? Of course, if you have a creek and there's rain and it's washing anything and it's seeping through the soil, which we call leaching, at the end of the day, it's going to end in the what? In the river. And you want to know whether the river is carrying it down and how far does it carry it. Should you be concerned? So periodically you do that. And so this is about I think it's about 4 or 5 years ago when my student went, and then I had another student go 2 years later. So this data is from a couple years, 2 to 3 years ago now. And what are you seeing in this data? What we are looking at, I want to tell you that the top is closest to the mine. You're seeing numbers CCS1 and all. Don't worry about that. Just remember this. The top samples, these are sample numbers you see in blue. The top samples are the closest sampling points to the mine, and the bottom ones will be farthest from the what? From the mine. And you can see somewhat there is a correlation where you have things seem to be higher, closer to the mine, and then you get to a point where I have said it's 22 MAG, and for some reason you can see that there is what? There's a resurgent. You see that? There's a resurgent, and this comes from some of the sediments that is near where the one of the creek is joining another. And so you not only sample the soil, we mainly concentrated in the solid. So we mainly sampled the soil around the place, but also the sediments. But you can see some of that is coming from the sediment, and you can see that that sediment amount, even just for copper, is way higher than the amount close to the mine. So that tells you there is what? There is mobility of those metals. And you can see it's reflected close when you go to something like zinc. Close to the mine, you have 2.82 parts per million. But when you go down to MAG, you have 8 parts per million. You see what I mean? And then same is reflected even when you go to the other side to manganese. It's almost double what it is. So what is that communicating to us? It's communicating to us that we need to pay attention to what is happening around that sampling site and alert the people around there that the mobility and transportation, there is accumulation there. And so the kind of activity that they should do there should be aware of the presence of these metals and their toxic nature. So I've only given you a cross-section of a lot of work that has been done in the Montana. It's ongoing. Part of what I do is I send students every year or every 2 years. They bring the samples, we check, and we monitor. Soil, sediments, and then I have, sometimes I've taken liquid. But liquid, you see, when you talk about liquid, the water is running. And chances that running water is you're going to catch it at that time are not as high as the sediment as it settles down. That is why we went there. But once or twice I've gone with the biology, and I do work with biology people, and a lot. And we went with them, and they looked at the limnology, and they picked also some of the insects and some of the life that was there. And what they do, then you go to the lab. Either they harvest some of the areas like the liver or the heart or wherever they think this could be lodged in, and they check that, and we correlate that to what we are seeing. So it's a beautiful kind of thing. And some of them look at the plants around the area, and they also harvest those plants, and then they look at the accumulation in the plants and compare it with the other sites and see whether what we are seeing tallies with what they're also seeing. Next slide, please. Phytoremediation, I talked about it. I just want to give you an idea of what we did with phytoremediation. The idea here is, can we use plants to uptake? And when you're talking about plants, you're not going to plant something that's going to grow for 100 years. You want to use fast-growing plants, right? Because you want to put it there, you want it to do the work, you want to harvest the plant, so that when you are harvesting this plant, you have also removed the heavy metal. And in this case, we were using kudzu, and I knew that kudzu may not, it's around, but it's not very common here as it's common in those areas. So that's the picture of the plant. But if you really want to know, you can also research, Google kudzu and see what plant that is. Now, how did we do this? We literally took kudzu plant, and we took it to the greenhouse, and we planted it under varying conditions of heavy metal, especially lead actually here, and we wanted to see how that stresses the plant. Now, I'm not biology heavy, okay? So when I make blunders with biology, just let it slide. But with plants, what happens when you stress them? They express that in terms of generation of what we call antioxidants, or actually the polyphenols. So you can actually measure that response or the elevation of the phenolic compounds in the plant will actually be a measure of the level of stress on the plant. Biologists, is this correct? And that's exactly what we did. So we exposed these, and then we measured the amount of lead and related that to the polyphenols. And what we realized is, so you have the plant, you have the leaves, you have the shoot, and you have the roots. And we realized for the kudzu plant that most of these was being lodged in the what? In the roots. So we realized that more in the root than the shoot, and it qualified as a rhizofiltrator. And so it really can be used to measure this. And what you're seeing here down on that graph there is just a snapshot of the concentration and how things. So when you add, EDTA is a binding agent, by the way. So we used EDTA to make sure the metal is bound there, and it's right on the place where we could use it. Right? It's almost like you're holding somebody, you want him to stay here. So when the metal wants that metal, it can find it. So that's why we used EDTA, bending agent. When you have no metal, you can see the amount of polyphenol is way low there. About 15 there in terms of, and then that's, we're using gallic acid, which is an example of polyphenol, to measure the level of polyphenols in there, and we're comparing that as a percentage of the control. Control is the one that didn't have any. So you can see when you have 0 metal, just a small level of polyphenol, if you may. When you have 100 parts per million of lead, you can see the rise in the amount of polyphenol as a register of how much the plant has taken in. And when you go to 200, you can see a large spike of that polyphenol. So that told us that, and we're analyzing this in the roots, and it told us that this plant can actually be able to uptake the lead and pre-concentrate it, if you may, in the roots. So it is a good plant that you can use. That work is quite some time back, and we put it in the Canadian Journal of Plant Science. And I can go on and on, But I think that we have many other speakers that are coming. So this is part of the environmental work I do. I have more work I do with soil. I have more work that I do in the environment. But I think this gives you a snapshot of what I do. So I can entertain some questions, please. Sure. I'm not looking for them, but I will take them. There's a hand here, and there's a hand back there. Thank you.
Thank you, Prof, for that important presentation. My question is, in Kenya we use a lot of fertilizers, and I have seen that in your presentation, fertilizers also contain these heavy metals.
Yes.
And now that fertilizers are containing heavy metals and we are using them, then this could be linked to the increased incidences of cancer, maybe in Kenya.
Yes.
Now, how can we determine the effect of fertilizer application vis-a-vis the increased cancer? Thank you. Maybe number two, also phytoremediation. Phytoremediation is one way, and if I got you well, there are three areas which you mentioned. Compost can be used to minimize these heavy metals. Another one is phytoremediation and use of tea waste. So which one could be the most effective in Kenya? Thank you.
Thank you very much. I want to say that I wish there was always a magic bullet, right? So that we can solve the problem once. So I'm starting with your second question. So to go with your second question, really, there is no magic bullet. So that there is no one way of those ways that we can use. However, a combination is always a good idea. So then what am I saying? So if I'm on that farm, I'm going to rely more on the manure. If I think that I have a factory that is coming up from the other side and there's a creek running at the bottom of my farm, I'm going to plant those I'm going to use phytoremediation plant, those plants there that are going to suck up this at that lower level. And of course, harvest them and take care of that. And now if I am having a lot of tea and I'm having tea waste or I know where I can find it, then I'm going to take that and on my, you need to mulch, right? When you're doing a garden or flower, you mulch. I'm going to use my tea waste as mulching for some of my garden, right? So that now with that mulching with the tea, it's not only going to give cover for the hard sun, but it's also going to help suck up any headache that is in there. So you see how it's not a magic bullet, but it's a way of renovating, and down the road, you do it once, you may not see the effect, but when you do it over time, you actually find out that you have something better. Let me go to your first question. I think I said this before in the talk that I did last time. So I grew up here, and I'll tell you, I left in my early 20s, and from the age of around 1 to the age of around 20, and I'm being very sincere, I heard of people have ugonjwa ya sukari. I never came into contact with one person. Never. I would hear of cancer, and it used to be a disease of the West, really. And it was very rare that I would, hear that and come across somebody who had that. From the age of 1 to about 20 years. That's a long time, right? Fast forward 2 decades plus, 3 decades later. I don't think there's a family that doesn't have somebody with diabetes. I don't think there's a family or clan that hasn't lost people to cancer. So, point that you're talking about here is why we have become more in population. We have found ways of making sure that we are growing things to do what? To eat. But at what price? So we are using what we call conventional fertilizers. And those conventional fertilizers give us a high yield quickly at the expense of something down the road. In fact, it's only about 3 years now that I was talking to my sisters. A good part of my family still lives here. And I was talking to my elder sister, and there's a farm that they were growing wimbi. And normally she would be like, okay, I need you to boost me so that I can bring the weeders. It's a labor-intensive job, right? To pull out those weeds. And every year I would give a boost financially to go do the work, right? That year she told me, don't worry about it this year. There is this magic spray that we are going to use. And I said, she didn't. I mean, your family doesn't register what you do, right? I don't think they register that I'm a chemist. And I said, what is this magic thing you use? And she says it's Roundup. And I mean, she blew my mind. And I said, you gonna spray Roundup on the farm? Yeah, I said, please. I'll still send you the money. Don't bring that any closer to the what? To the plants. Now, yes, it's magically gonna kill the weeds. At what price? So that is what's happening. So some of the things that we are using, like those Roundup, they are banned in some of the Western countries for use in products that you're gonna eat like that. And so, but we are using them because our regulation has not caught up. It's going to catch up when we are overwhelmed with the cancer cases. Now, when you use Roundup right now, find DDT in the stores. It's been banned for decades in the US. I mean, it's like maybe 40 years now banned. And you can still find it. So when you use these things, you're not going to get sick immediately. It's going to take 8 to 10 years. And then you start having an increase in the cases of cancer and all that diseases, and then you wonder why. So this is the answer to your question. So how do we do it? Politics kind of runs everything. If we don't have good regulation, it doesn't matter how good science we do. So in the US, when you are an environmental polluter, the fines are very steep. Very, very steep fines. So then the people have to think twice before they do it. Here, I don't know if we have caught up with the fines. There was another question in the back, please. Yes, 2 hands in the back.
Yeah, thanks, Prof, for the wonderful presentation. You have done exceptionally well to share insights, especially in terms of the research and contribution that you've undertaken. The exciting part of it is, to me, is that aspect of heavy metals. I was wondering if the same can be extrapolated to extraction of some of these precious metals from electronic wastes, so that we can find some ways to circumvent, for example, the recent China policy on exporting their precious metals to other countries.
Very good question.
And then another one, just to sort of benchmark on what you have done with the Kisii plant. You've sort of monitored your river for some time. I don't know whether you've built a database that can be used as an information portal, so that we can borrow from your side on what you've done. Thank you.
Thank you. So I'll start with your last question again. Yes, we have a lot of data from the Montana. In fact, they were going there before I came to University of Laverne, and I have not. You almost caught me when I came in. There was a lot of movement from physical to digital. I have not caught up. In fact, I started with thesis, so they had a lot of thesis where every just like here. I hope you. My mind goes if you guys do that, but every student who graduates with chemistry has to do a thesis project, and they write a thesis. But they had all these, and I have them in my office, at the chair's office. I have all these bound theses, and I told them nobody's looking at this stuff, so let's turn these into electronics, so at least somebody can search, and other students, and so they can pick up on that. So we started with that, and then when I went to the Montana project, I realized there's a lot of data that has come. We haven't caught up, and usually I have to find a student worker that I have to do this. I had one from computer science that did a wonderful job, but it's a challenge for me. In fact, it's a challenge in that context, and me and you had that conversation. We need to get to the point where, if the situation allows, we can put a monitor there that sends information to us right away, so that we are able to do what? To monitor effectively without having to wait with a lag. Okay? Remote. First question, precious metals from electronics. Yeah, it's a grand idea. In fact, you know that in I think it's in Southern Korea, they take this what we don't use here now. We used to use a lot of plastic bags, right? So they will take this plastic bag, and that technology is there, and you burn it, and when you burn it, you get the gas or whatever is there. You can use that for for energy, but also the product that's coming out there to another, material or another product, so that you are not wasting them now, you are reusing them. We need to be able to get to the point for the electronics too, because it shouldn't. It depends on what is the major component. They either thermosetting or thermosoftening. So the polymer will either harden with heat or soften with heat, because heat is usually what we do. The only challenge we have when you are going to use heat to harvest these precious metals from there, you see, you have to think green. The way to think green is, if I'm using heat, I'm going to generate some what? Volatiles, right? How do I put what we call scrubbers or filters so that doesn't go into the atmosphere? That's the challenge, number one. Number two, how much energy is it going to need for me to do that? You want to be able to be energy efficient while you are trying to recycle the material. The second thing you ask yourself is, if I'm not going to use heat, then I'm going to use chemicals. What chemicals am I going to use? If I use acids, what do I do with them and with whatever they will eat from the plastic? So that's the challenge. So we have to think green chemistry is a great idea. We have to think, when we don't think, the guy who is interested in money is going to do the job. And because they're interested in making money, not in the environment, they're going to do it in the crude way, as they always do. They're going to just burn it and release the toxins to the environment. And it's you and me wondering what happened. Or they're going to use the harsh chemicals, and they're going to dispense those to the creek. So that's a challenge question for us. Thanks for asking that. Is there any other question?
Yes, thank you very much.
Yes,
Prof.
please.
Yes, you said kudzu can be used for phytoremediation.
Yes.
It's a plant.
Kudzu is a plant, yeah. There
So
was a picture there.
Yes. What are the ecological implications if we are to use it for a large-scale effort to do phytoremediation?
Thank you. It's always about the environmental eco-balance, right? So let me take the example of Lake Victoria. We know we have what? Is it called the hyacinth? What is that plant called? What a hyacinth. So there is pluses and minuses of that plant over there. It gives the cover. It serves as food for some of those lives there. But then, on the other hand, what else is it doing? So then it's always a balance between, if I'm going to harvest it, what am I going to do with it at the end of the day? So when you look at phytoremediators like kudzu plant, you have a super fund place. So you look at, if I have mercury, I have lead, I have arsenic, I have chromium, which can get to chromium 6 and become very toxic, right? Then you have this phytoremediator that can remove it. Then the first thing to do is, yes, let me remove it. Because where it is, I cannot control it, right? Now, once you have removed it, you have it in the plant, your next problem comes, what do I do with the plant? And I think that was your question, right? So then you come into the science and say, incinerators, burning it, is probably one of the ways you can do that, right? And if you do that, your incinerator needs to have scrubbers and filters for those gases that are being released. And that becomes the way to do it. I mean, you know, all the time, you have heard of chemical warfare, right? So we have chemicals that we used to do those things years back. They have incinerated most of these, and they do it that way. So what you're always looking for is a way to control an almost running situation, because when it's out there, you can't control. If I release a gas out here, it depends on the way the wind is blowing. Now I have no control. But if I have a way of scrubbing it in, just like we do for carbon dioxide, and we trap it, and we say we have done some carbon whatever, that through the plants and through many other ways, that is what we are looking for. Thank you. Question?
Thank you, Prof.
Yes.
My question is very simple, but I have to ask it.
Please.
You indicated that whenever we use these chemicals in the environment, it's like they create much more harm than good, especially to our food. Now my big worry is, how do we then marry technology and food production?
That is the elephant in the room. They don't pay me enough to dig the answer, but so before we advanced in technology, the first person who really manufactured the car, people were like, you're crazy. You ain't going to have any movement without what? Without a horse or a donkey. And when this thing came out, and the guy and his wife, this was Ford, by the way, in Michigan, everybody was baffled. So this thing this guy has been doing in the warehouse is really something, and it can move, and you don't have to have an animal pulling it, right? So now we are advancing in technology. We even have what we call nanotechnology. It's not that we have invented it. It was just there, but we had not gotten to the point of harvesting. Because what nano is everything grows from that scale down there, right? So if I can weave it at that point, right, I can make the material stronger. I can make the material more soluble. I can make the material more, I can put functional groups that can make it bind to this and bind to that. You see where I'm going. So as we increase in technology, there are things with that technology that become beneficial to us. I think we need to lessen down on the economics. What usually ruins things is the gluttony of it. Because when we discovered plastics are not evil per se. But whenever, America is the worst in terms of how we use things. We are sometimes the largest polluters. So when plastics came, everything became plastic. That is the problem. So in our farming, in our wanting to feed people, let's not be driven by the economics. Because when we are driven by the economics, then it is like, I need to give it much more nitrogen, much more phosphorus, much more that, so that I can produce more, make more money. Am I speaking? So I think if we have a balance, and many times, and I'm speaking from ignorance when it comes to Kenya, but in many places, many times, the scientist is not in the room when these decisions are being made. So if the scientist can be in the room, then we'll, I'll give you an example of why I say that. Long time ago, I worked with the Department of Water, and I was in the headquarters. And Nakuru was a growing city, and they needed water. And I think it's the Japanese or somebody was contracted to bring water from, I don't know what, one of those rivers on the other side. It was several kilometers, hundreds of kilometers away. The pipe that was bringing the water, I went there and I can walk inside the pipe. That gives you the size of the pipe, right? So all that water is coming and it's good for the city. But then there was no science in the room to say, once we bring this water, people are going to use it. Where does it go? And so what happens is the water comes, people had the water, they used it, and we fed it to the lake, right? We have a lake right there. What happens? There was a lot of nutrients, a lot of eutrophication, because, think about all the detergents and everything, the phosphorus and all that, right? And there was a lot of eutrophication, and that meant that it's wonderful. There is food for the fish and all that. But then when those plants are going to decay, they use up all the oxygen. They choked the lake. The lake started dying. The president then, I think, was Moi, and then he told the scientists, they warned him before that, but he said, no, you just want this city where I live really to look bad. Now, when the lake started dying and the flamingos were going away, he came back and said, you just want to sabotage me. So you see, that's the problem here. The scientist is never in the room to give an idea, a balanced idea of how we do this. So yes, we don't have to shun technology, just like we can't shun AI, but we have to manage AI. I mean, if you are a professor of English composition, for example, essay, and you tell your students you can use whatever you want, I'll tell AI right now to write a composition about Kisii University. It will be more beautiful than what I can write in. They will do that in 2 minutes, and I'll have to take 2 hours to do it. You see what I'm saying? Can we shun it? No. Should we manage it? Yes. Yes, please. Question here, question there. How much time do I have? You have to control. I don't know. I don't know time. Yeah.
First, I want to thank you so much for the insightful discussion we're actually having right now. And I'm sorry that I came a little bit late. Was caught up, was traveling from Nairobi. So my apologies.
Welcome.
Yeah, I think I caught your discussion a little bit late, and it's really interesting. And I'm marrying this aspect of technology that has already been raised.
Mm-hmm.
And so my question, maybe I have possibly 3 questions that I may want you to help us. So one of our MSc students actually did VAMI technology, and we really wanted to extend that to the community. So possibly, how can can we be able to, come up with something that can be able to extend that technology to the farmers, because it's purely organic. From the vermi liquid, you are able to get a pesticide, and from the vermi that you are actually getting, you're using as fertilizer. 2 is, as a biotechnologist, I'm interested. For example, you said that there are some compounds that you realized are able to remove contaminants from the environment. So maybe a quick one that I can be able to raise is, you went ahead and did tissue culture, right? For mass cultivation.
Okay.
Right.
Tissue culture.
Yes.
Okay.
So as a biotechnologist, I could be interested, like, for example, how can I minimize that particular plant, but then I'm able to remove more, of the toxicants from the environment. So is there any study that has been carried out, trying to upgrade that particular gene for that, responsible for removing that? Then lastly is, I'm glad you also talked about nano, which is also my area. So what studies that have been carried along that side, your specific plant of interest, or is there something that we can, come up together with? Thank you.
So, the answers to your questions are not much in that connection of driving it through the kudzu plant. but it's the answer that addresses me and you, those who have PhDs. One of the things you realize with a PhD is that you know too much about too little. Did you learn that? You know too much about what? Too little. So the only way the PhDs make sense is now when we team up and I say, okay, you know too much on this area and I know too much here. Can we put it together? So in answer to your question, yeah, we worked on this line of the kudzu and on nanotechnology so maybe yes, we can put brains together and look at that. Now when it comes to technology and farmers, a lot of strides have been made, but I think we are only still scratching the surface. Okay, so there is a lot more work that needs to be done, but if that work is going to be meaningful, it has to take the scientists, the farmer, and the interested community. Because as you have seen it in terms of tourism and in terms of some of the farmers who have the ability to be commercial, they have used the remote sensors and whatever to check on what is happening to their plants. And that speaks to, I'm seeing that is happening to this side of without being in the farm. And now it speaks to them and they can now go and do that. So yes, a lot can be done in that area. It doesn't just have to be only in sensing. But it has to take this person knowing too much here, that one knowing too much about too little, put the heads together and get the work going.
Thank you.
Of course, if we don't engage the farmer, just like the first speaker said, it will remain in the publications.
Gathering dust.
Yes. All right.
Okay, thank you, members. I think because of time,
yes,
we can stop there so that at least we can go to the next session. So otherwise, Prof, I think I thank you so much for the nice presentation, and it's good. I think members, we've really gotten something. Let's give him a hearty clap. One. Thank you, thank you, thank you.
So I hope I'll get a minute later on to show 2 slides about opportunities that may be available for faculty, but I want to do that at the end.
Okay, that is no problem.
And I'm holding that so that the faculty can stay behind.
Okay.
Thank you.
Okay, members, I think because of time, as I said, and I can see tea is ready, I would like us, first of all, before we go for tea, we can have one presentation. And according to what I have here, I hope Dr. Eric Mobegi, you are ready. You can do the presentation. Then we have about 10 minutes presentation, then question time, then we go for tea. Welcome.
Good morning, everyone. My name is Mobegi. I'm working on, or I've been working on, application of nanotechnology for low-cost point-of-use filters, and we are targeting 2 pollutants, that is arsenic and fluoride, and mostly this is with respect to underground water. Next. Now, this is how we're going to go through that presentation. We'll deal with the overview of water scarcity as it is now. Then we'll look at water quality and pollution. What are some of the interventions that have been made in terms of water treatment technologies? Then we'll also see innovations which have been carried through. Then we will delve into our research. Now, currently, when we look at the water distribution in our world today, we find that 97.5 is just but salt water. Surface water only accounts for about 0.4%. That includes our rivers, lakes. Then we have got underground water, which is about 30.1%. But when we look at the statistics, we find out that only 0.5% of water on earth is usable and available in terms of fresh water. So you can see the crisis that we are in. Now, this supply of 0.5% is getting depleted at a higher or alarming rate because of what? Climate change. It's something now that we have to deal with currently. Look 20 years ago. Where were we in terms of water? You could go to the spring, drink your water comfortably, go back home. But it's not something that you can dare do right now. Next. Now, globally, there are about 2 billion people who do not have access to safe drinking water. That is not a photoshopped picture. It is a real picture of a young man directly drinking from dirty water. That should be in some place in Turkana. Next. Now, when we look at the scenario in Africa, where are we? Actually, they are talking that by year 2025, where we are in this year, water scarcity will be what? Very high. But also when we look at the distribution worldwide, countries which are now being considered what? Waterless. Kenya stands at 2.27%. It is something to get us worried. But as scientists, we can always what? Offer a solution. Now, which is some of these causes of water scarcity? Number one is about population growth, climate change, and industrialization. Having this in the background, next slide, there is that ongoing water shift. We are now moving from surface water because of its pollution to underground water. But how safe is this underground water? We'll be looking at it shortly. Now, when we look at water quality, we'll look at the three aspects: the chemical properties, the biological properties, and the physical properties. Mostly, I'll look at the chemical, where we'll be dealing with metals. I think Prof had mentioned about it, especially heavy metals, pesticides, and other organic compounds, which came through during the plenary. Again, there are some other physical properties which affect the quality of water. For example, color, turbidity, etc. Because the reason why we have got this is our research is going to revolve around the chemical and physical properties. Next. Now, what are these consequences associated with contaminated water? You can look at a number of what? Diseases. But yet, we are yet to see the worst. After moving, mostly this is surface water that leads to these diseases. But when we move to underground water, we're yet to see the worst. Next. Now, since surface water is increasingly becoming polluted, we have resolved to underground water. And more of this is being done by sinking boreholes, day in, day out. Not knowing that even we are doing what? We are affecting the stability of what? Our earth. They are saying London is a sinking what? City, for those who have traveled wildly. Because every day we are sinking a borehole. And yet we have been given resources which we can be able to use surface water than going for what? Underground water. One of the three main concerns of underground water are three pollutants. Among others, we have fluoride, we have got arsenic, and mercury. Next slide. Byron, next. Okay. Now, look at the health effects of fluoride. This is nowhere outside Kenya. This is Nakuru County. And normally, the WHO gives a limit of 1.5 parts per million, which is safe for human consumption. But in other areas in this country, levels above this set limit have been found, and mostly along the Rift Valley belt. It has led to two major diseases, that is dental and skeletal fluorosis. Now, I know we know this guy, Robert Alai, is currently the MC for which ward? Is it Kileleshwa? Nairobi. This is what he said. Hey Alai, I'm a dental surgeon in Nairobi. My practice is actually in Kileleshwa off Riverside Drive. The number of kids we are seeing with dental fluorosis, the brown teeth, in Kile due to what? High levels of fluoride in borehole water is crazy. That is in Nairobi. And if we have to follow the WARMA regulations, we are not supposed to sink a borehole in a radius of what? Is it 2 or 3 kilometers? But you find somebody has a borehole here, the next neighbor has what? A borehole. Still compromising on that. Now, statistics show that 19 million Kenyans suffer from what? Fluorosis. Now you see mimi nimesema Kenya Society for what? Fluoride Research. Borehole water from Gilgil, a research done in 2011 by Wambu and colleagues, showed 6.5 ppm. Very dangerous. Then boreholes from water in Nairobi, because of the aquifer there, it has been found to be 15 ppm. Extremely dangerous. And once you suffer from what? This fluorosis, be sure it's a condition which is irreversible. Right? Now, other than fluoride in underground water, another emerging contaminant is what? Arsenic. WHO requires that this is supposed to be less than 10 parts per billion, and any level above that, and with accumulated use, it is dangerous to what? Human health, because it causes cancer of the skin, cancer of the liver, among other types of what? Cancer. This is, go back. This is a case I saw myself in India. Now, is it affecting us currently? Yes. Next slide. Byron, next slide. Look, this is in Kenya. This was a story done by one famous journalist known as Dennis Onsarigo, in a story entitled Desert of Death. Go take your time and look for it in YouTube. Desert of Death. When he was doing this story, this kid, ukiangalia hiyo, you may not even eat. He died 3 days later because he had suffered from what? Cancer. The same thing with that lady. No, go back. The same thing with that lady. This is in the northern part of what? Kenya, Marsabit County to be what? Specific, a place called Kargi. Next. Now, some selected boreholes in Nairobi have shown to have these high levels. Levels ranging between 4.5 parts per billion all the way in some lakes in East African Rift Valley to consist 22.7 ppb. Very dangerous. Next. Now, what are some of these intervention technology that has been there? For example, we have a technology such as what? Reverse osmosis. Reverse osmosis cannot be able to remove arsenic for us, but it can remove fluoride for us. But how many people can afford what? Reverse osmosis. And there are limitations associated with reverse osmosis. Kuna imagine tumezoea kukunywa in the name of being packaged. A lot of that water does not have the minerals that the body requires, because the reverse or the membranes in the reverse osmosis has extracted and the water has become flat. For fluoride, commonly used is what? Bone char. And we are going to see examples of that. Next, let's move next. So that is reverse osmosis. I've mentioned about it. Next. Now, this is the production of what? Bone char. This is done by the Catholic Diocese of Nakuru. But there are associated problems when we use bone char. Number one is the discoloration of what? Water. It turns brown and it has got what? Smell. There are people even who have used donkey bones. And if somebody knows that you are using donkey bone, can he take that water? He can't. Next. So that bone char is packaged into what we call a terrafill candle. Then it's used to remove what? Fluoride. It's cheap, but I've also explained the disadvantages. Next. So chlorination, especially for biological contamination, is good. But again, it comes with its disadvantages. Formation of what? THMS, such as what? Chloroform. And this is one of the compounds which has been associated with cancer. You know, in the village, when they are on a pair of zile tablets, they always imagine if they put more of it in the water, it is when the water is what? Safe. Not knowing they are exposing themselves what? To diseases. And these THMS have been found to cause bladder and rectal what? Cancer. Could it be one of the cases where we're having surge in cancer cases in the country? Next. So UV Prof mentioned about it. Yeah, but still it's unaffordable to most of the people because of the poverty levels. Next. So there's an innovation which has been done by a company known as Chujio. Mostly this can only deal with microbial decontamination using colloidal silver. But again, it's not affordable to the poor in our society. Actually, they sell them through NGOs such as World Vision. Next. Now, this is one of the nanotechnology-based solution. This has been done by a guy from Nelson Mandela Institute of Science and Technology in Tanzania. It's known as the Gongali model. This is the guy who is in a suit and is using nano particles, package them in a what? In a terrafill, then it's able to simultaneously remove fluoride, bacteria, and amoeba. Still, this one, the cost is not as cheap as it's supposed to be for most people. Next. Now, this one has been done by, it's called Amrit. It has been used for arsenic removal. And this is where the lab I was in that developed this innovation. It is able to remove arsenic, and it has got kinetics in terms of seconds, reducing from 300 parts per billion to less than 1 parts per billion. This professor, which I'm going to show you the last part of my slides, has been contracted by the government of India to supply all this. As of 2017, he had not even reached 20% of that contract. And remember, we are talking about innovation from science. Next. Now, what is the big question in terms of what? Improving our water quality. The solution now, go back. The solution now lies in the tapping of nanotechnology. And when we talk of nanotechnology, we are just manipulating matter to the size of less than what? 100 nanometers. What have we done? Next slide. Nanomaterials have been used for photocatalysts, that is to break down, for example, dyes, which can contaminate wastewater. We can use, for example, silver and gold nanoparticles for disinfection. We can use nanofiltration, like a good example I'm going to show you. And also, we can deal with what? Nanoadsorption. Next. Now, in this nanotechnology, we can deal with 0-dimension to 3-dimension nano-based materials. But in our research, we are focused more on using what? Zero dimension. Mostly using metal nanoparticles. And when we look at 2D, somebody mentioned about pesticides and other persistent organic pollutants. I think Dr. Where is he? Rono. Graphene. We're using reduced graphene to remove pesticides from what? From water. Next. Now, in terms of the research that we did, we were using a biopolymer mixed with nanoparticles of aluminium, iron, titanium, and zirconium. Each played a key role. I remember, Prof, you mentioned about the strength of the materials. For example, zirconium was helping to harden this material. Now, from the lab, we were able to move to continuous flow mode studies, which is going to give us an innovation of a point of use filter. Then thereafter, we'll deal with what? The safety. Yes, we have developed the material in the lab, but how safe it is. We don't want to solve one problem and create what? Another problem. Next. So that is a scanning electron microscope micrograph showing images of granulated material that now we pack in this filter. And you can see that after even running the contaminant through it, it maintains its strength, because nothing changes on the surface. And one thing that we look at are those adsorbents is that it must maintain its surface integrity. Actually, those grains, hata ukiziweka kwa maji, they don't dissolve. Next. So this is a high resolution transmission electron microscope. And if you look at magnification of 5 nanometers, that is at C, you can see some black spots, which shows some embedment of what? Nanoparticles, but mostly is what? Polycrystalline. Next. Now, this is still a micrograph for high resolution transmission electron microscope before adsorption and after adsorption, showing us, is there adsorption taking place? Yes. This is FTIR. You can see that at about, after adsorption for fluoride, the peak at 1107 disappears. And when we move to arsenate, there are 2 prod beaks, which appear, the red one after adsorption, which also shows that arsenic was what? Adsorbed. But mostly this kind of adsorption is physical and not chemical. Next. Now, when you are working with a solution to deal with water purification, pH plays an important role. But remember, drinking water, the pH ranges between 6.5 to 8.5. It's not possible to find the pH of drinking water at 7. If you see that in that packaged bottle, run away from that water. Because water can never be neutral, right? So the pH, if you look at that material, it can work across all the pH ranges. And effectively between 6 and 8, it's good to do what? To do that, even for arsenate. But we will see that for arsenate, it declines in the basic what? Region, and is so good in the acidic what? Region. Next. Then one other factor that you have to consider when you are fabricating such a filter is the effect of competing ions. What do we mean by that? Those ions can be either positively charged or negatively charged. So they will compete for the active adsorption what? Site or binding site. If the competition is higher than the contaminant that you're looking at, then it becomes what? Not viable for commercialization. However, there are some, because they coexist with the other ions in water, for example, chloride, silicates, hydrogen carbonate, and carbonates. You'll see that they play what? A key role. But you will see that definitely is that chlorides for fluoride, they tend to compete very high. The same for what? Silicates, carbonates, and hydrogen carbonates when dealing with what? Arsenate. Because of a simple reason, they compete for the active site. Next. This is, if you can begin from an initial concentration, it can show you. For example, it's difficult to find 100 ppb of fluoride, but still, you can see the percentage reduction as you increase the concentration for both fluoride and arsenate. Next. Now, once you've synthesized this material, how long can you use it? And that is when we deal with what? Regeneration studies. For example, for fluoride, you can use that material 4 times. And this in terms of milligrams. But when we'll be going to the prototype filter, we'll deal with the grams. But for arsenic, you can go up to 6 cycles before its efficacy reduces to lower than 90%. Next. Now, this is where we started doing our prototype point of use nanofilter. Look at the water in the upper beaker. It is highly turbid and yellow, or slightly yellow. But after going through the filter, the black things that you are seeing on the right-hand side, they are packaged in that cartridge. That cartridge is fixed here on a brick block, which enables to tap other particles which might affect what? The material. Next. This is now the design of the final product. And we can use this in our homes, and that's why we say point of what? Use. Just get your water, run it, and you're able to use it. Next. Now, once we developed that prototype, we found out that it can be able, with only 60 grams of that material, we can reduce fluoride from 10 ppb, where we have got the input, to less than 1.5 of 275. This is what? 60 milligrams. And for arsenic, we can deal with what? We can reduce 650 liters before we hit the WHO limit. So this shows that the material is extremely viable. I'm seeing time's up and seek for your indulgence. Next. Now, how does this filter perform with other what? Parameters. You can see it is able to reduce fluoride from 6.6 to 1.3, which is less than the recommended. Then turbidity is able to reduce from 0.9 to 0. Next. Then other water quality parameters. You find that color, which affects the attitude of the consumer, can be able to reduce from 24 to 0. Turbidity from 150 to about 0.6. Next. Actually, this was a sample which was picked from Gilgil, where we have got high fluoride. The safety of the material is checked against leaching. Professor, you mentioned about this. And all the metals that we use for the synthesis of the nano adsorbent were all less than the limit set by WHO. What does this mean? It can provide safe and clean drinking water upon what? The reduction of these contaminants. Next. What about when this material you've used over and again, you have to deposit somewhere, for example, a landfill. You have to do a test known as leaching experiment for total organic carbon. And normally the WHO states that all the materials used to synthesize that must be less than 4 ppm. And you can see for both fluoride and arsenate are less than what? 4 ppm. Next. Now, where are we on with this research? We are in the process of registering the patent. Once we get that patent, now we move to upscaling, and then now we will go the commercial way. Next. Now, Professor was talking